Okay, so it's our great pleasure to receive Dr. Carlo Freire Oliveira. So Dr. Carlo Oliveira is an old friend of us here, of everybody, I guess, from São Paulo, Rio, and Uberlândia. And Carlo did his master's and PhD at Ribeirão Preto Medical School and University of São Paulo in basic and applied immunology, where he spent some time at NIH in Dr. Marcos, José Marcos Ribeiro's lab, always working with saliva from vectors. I remember when I joined the lab for my master's, Carlo had several of these sticks stuck into an adhesive tape collecting saliva, as you can see in the picture here. So it's a great pleasure to receive you here today. He's a professor at the Federal University of Uberaba, and he's going to present his talk entitled Anti-cytokine Activity in the Saliva of Brazilian Ticks. Thank you very much, Carlo.
First of all, I would like to thank for being invited to this meeting. This webinar is very important for us to share our knowledge we produce here in Uberaba, Minas Gerais. And today I'm talking about anti-cytokine activity in the saliva of Brazilian ticks. My name is Carlo without S, Carlo. I'm head of the lab of immunology and bioinformatics. I work at Federal University of Triângulo Mineiro, here in Uberaba, Minas Gerais. And I would like to thank to all the guests, hosts, and organizers of this talk. And today I'm going to talk something that is very important in my career, because I always worked with ticks and cytokines. And before I connect all the information when ticks and cytokines, I'd like to make some brief presentation, introduction about these protagonists, the cytokines and ticks, just to get everybody involved with the subject of the presentation. So ticks, here in Brazil, we call ticks as carrapatos or chatos. And Spanish-speaking countries, they call ticks as carraças or carapatas. And ticks, they are hematophagous ectoparasite arthropods that infest mammals and birds. And in a lesser extension, they are also able to infest reptiles and amphibians. They cause direct damage through infestation. This is very important, economically speaking, but they are also important because they transmit a lot of disease. Here in Brazil, we can say Brazilian spot fever, ehrlichiosis, babesiosis, and Lyme disease in USA is very important in that country. And the ticks, they climb on grass and bushes and trees and infest the skin. After that, they inoculate the buccal apparatus in the skin and inject saliva in the host to modulate the immune response and hemostasis. Ticks, there are a lot of ticks around the world. They are from the Amalia kingdom. They have articulated legs. They are from the Arthroptophilum and have a lot of families in ticks. They are from the Ixodidae, Argazidae, or Nutalidae family. There are a lot of species of ticks. Globally, approximately 950 species of ticks in neotropical regions like South America, Central America, nearly 200 tick species. Only in Brazil, there are 70 species of ticks, but most of them are from wide environment. And here in Brazil, the most important tick species are Rhipicephalus sanguineus, the broad dog tick, Rhipicephalus boethus micropus, the cattle tick, and Amblyomma sculptum from the Amblyomma carinense complex, the rose tick. The rose tick is very important because they transmit a lot of disease in Brazil, and they are able to infest also, besides horses, they are also able to infest dogs, capybaras, and humans. That's why they transmit disease to humans. In Brazil, the most important disease transmitted by Amblyomys sculptum is Brazilian spot fever. So we know about ticks, and when we try to understand how ticks infest the host, it's very important to understand how we collect, how they produce the saliva, and how we can get the saliva to make the experiments. So we can mimic the natural infestation using lab animals like dogs, rabbits, and guinea pigs. We put the animals in the animal's back, you put ticks in the animal's back, and after that, during the cycle, you can put larvae, nymphs, and adults. And when they get feeding, when they feeding, they fall, and we take these animals, inject dopamine, and inducing salivation. You inject dopamine in celomic cavity, induce salivation, you take the saliva, go to the lab, and make the experiments. And these experiments depends the amount of saliva we can collect during the natural infestation, but we can use natural infestations, artificial tick feeding, and the infestation in labs with rabbits, guinea pigs, and dogs. So we know about ticks. They are hematophagic arthropods. They are very important to transmit a lot of disease, and they modulate the hosts by producing molecules that are present in the saliva. And they inject the saliva with these compounds. There are modulation of the host homeostasis and immune responses. So today, I would like to, everybody who knows about cytokines, but I would like only to reinforce that for us that working with cytokines, I'd like to demonstrate the main information about that cytokines are peptides that are the mass regulator of the immune response, from the immunological development to the different stage of the immunity, including resolution of the immune responses. And I believe, as a scientist, that if tick wants to get some success in the infestation during the feeding, if they want to be successful in the feeding for many weeks, sometimes 3 weeks, like buffalo microbe that stay in the host for 3 weeks, they need to modulate many mechanism of the host defense. So for us, cytokine is a strategic way to modulate to get successful in the infestation. So cells after stimulus induce cytokine production like TNF, interferon gamma, and IL-4, IL-6. And after that, after secretion of these molecules, they act on all the different cells and exert the biological effects. There are a lot of effects from the cytokines like proliferation, cell growth, activation, immunomodulation. So there are a lot of mechanisms why cytokines modulate the host immunity. And I think that ticks is able to modulate these molecules to get successful during the parasitism. So the first question is, against who the ticks are defend themselves? They can defend themselves against bacteria, viruses, fungi, parasites, vertebrate host defenses, like vasoconstriction, platelet aggregation, coagulation, inflammation, and immune responses. Bacteria, viruses, and fungus and parasites, they are very, very important to the health of these ticks. So ticks defend themselves by using cells and molecules present in many different tissues: hemolymph, body fat, intestine, and salivary glands. For me, the molecules that are produced by hemolymph, body fat, and intestine are related to defense against bacteria, viruses, fungus, and parasites. But the most of the molecules that are produced by salivary glands are related with the immune response against the host immunity. That's why most of the markers that we isolated and made the identification were produced by the saliva, by the salivary glands. So to have an idea how many molecules are produced by saliva of ticks, I made a very clear investigation in the Medline. In the 70s, only 2 molecules were described and identified. In the 80s, 1 molecule only. In the 90s, 6 molecules were identified and characterized in the saliva of ticks. After that, in the 20s, 38 molecules, and in the last decade, 25 molecules were identified and characterized in the saliva of ticks. This is nice because in the first decades, only 1 molecule were identified, but in the last 25 years, the number is increasing a lot because there are a lot of many technological methodologies that were used, like proteome, transcriptomes, and other biochemical investigation that was possible to identify these molecules. Last month, I just published a manuscript sharing which kind of tick is producing these molecules. Most of the studies demonstrated that saliva from the tick family, Amaphysalis, Amblyomma, Ornithodoros, and Argus were the most important family of ticks that molecules were produced and identified and characterized. And so, as I said, until now, I demonstrate that ticks are important because they cause direct damage and they transmit disease. And I said also that one of the most important things that is used by ticks to modulate the immune response are the molecules produced by the saliva. And we published a manuscript 5 years ago with Anderson Sanunes and José Marcos Ribeiro. We published a review that is very, very nice. And we published that to demonstrate some evidence of arms races in the coevolution of blood-feed arthropods and vertebrate hosts. And we demonstrate that there are many phenotypes that are increased, exuberant, in blood-feed arthropods and vertebrate hosts, as a fight that exists between them along of many, many millions of years. So this adaptation, this coevolution between ticks and other blood-feed arthropods and the vertebrate hosts involve anatomical, molecular, and gene changes, both in the blood-feeding arthropods like ticks and vertebrate hosts. For example, blood-feed arthropods has a very big salivary gland, and the host has a lot of mast cells and basophils, 2 of the most important cells to protect the host against ticks. In a molecular level, they produce a lot of molecules that modulates molecules from the host, like modulation of histamine, serotonin, leukotrienes, IgA antibodies, and complement system, proteins of the complement system. In a gene level, they have a lot of molecules that genes related to molecules that modulate the immune response. The same thing happens to the host, like mammals. So this is a very nice publication that we did it in Trends in Parasitology 5 years ago. So the first perception at first glance, I always studied the saliva, molecules produced by saliva in a cellular perspective. It was not a problem, but at the first glance, I thought, maybe ticks must modulate innate and adaptive immune cells. This is right, but it's not enough. We published a lot of manuscripts our group published a lot of manuscripts showing that saliva, molecules from saliva, are able to modulate macrophages, dendritic cells, eosinophils, neutrophils, basophils, B cells, CD4, CD8 cells. And we published this demonstrating many molecules that are produced, modulate the cells, allostatins, prostaglandins. They modulate differentiation, activation, antigen presentation, and induction of pattern of immune responses. This is nice. 1 month ago, we published a chapter book demonstrate all the molecules that are able to modulate cells, not molecules, but cells of the immune responses. So if you see this table, we see a lot of molecules like prostaglandin, dopamine, adenosine, allostatin, many other molecules that modulate cells, differentiation, activation, cytokine production, and many other functions of these cells. But the immune responses is more than that. We have a lot of cells, and all of them is modulated for some molecule from the saliva. But the immune response, as I said at the beginning, cytokines like TNF, interferon gamma, and many others are central, are key regulator of the immunity. So in the last year, I'm trying to understand the molecules from the saliva in a molecular perspective, how these molecules interact with cytokines, chemokines, antibodies, to understand if the tick molecules are able to modulate molecular for the immunity. So I'm in this presentation, I'm moving to explain how ticks modulate chemokines and cytokines. Chemokines is a kind of cytokine with chemoattractant functions. So the first experiment that I did was during my master's degree. And I did a very simple experiment of cell migration using Boyd chambers. Before, when I I add chemokine here, cells from this compartment migrate to the chemokine. When we co-incubate saliva with the chemokine MIP1 alpha, the saliva in different concentrations inhibits the capacity of MIP1 alpha in attract dendritic cells. This was the first experiment that I did during my master degree. I was looking for the mechanism why tick modulates cells. But during this experiment, I changed my mind and I said, let's see what happens if I put the saliva of ticks in molecules. And I was trying to understand dendritic cell migration. And it was not in my a specific objective of my master degree, but even so, I did it. When I put saliva with the chemokine MIP1 alpha, 1 alpha lost the capacity of attracting dendritic cells. Saliva per se does not have this kind of function, modulate attraction of cells, inhibiting or activate cell migration. But when I put the saliva with the chemokine lost the capacity to attract dendritic cells. So the effect is on the chemokine, not to the cell. And I demonstrate that this effect is target specific because when I co-incubate saliva with other chemokines like HANTs, MCP1, and SDF1 alpha, the saliva does not have effect in inhibiting the attraction of dendritic cells. So I demonstrate for the first time that saliva from Rhipicephalus sanguinea, the dog tick, is able to modulate specifically MIP1 alpha. And here I'm demonstrating this in this graph here, the inhibition of the migration inhibition that is happens in a concentration dependent manner. If I increase the concentration of the saliva in the co-incubation of the chemokine, the MIP1 alpha decreases the capacity of attract dendritic cells. This was very nice. But the group of Amanda Proudfoot published with the collaboration of my advisor during master degree, Beatriz Rossetti Ferreira. I have not part of this, but they published that ticks produce a family of molecules that bind to chemokines. They produce a family, a few dozen of molecules from the same family they call avasins. And these molecules are able to produce highly selective chemokine binding proteins with anti-inflammatory activities in vivo in many inflammatory models of immune-mediated disease. So they describe in this publication evasin 1 that binds to MIP1 alpha, MIP1 beta, and PARC, evasin 3 that binds to IL-8 and GRO alpha, and evasin 4 that binds to MIP1 alpha, RHANTES, and L-taxin. In present days, we know that in fact, there are a lot of avasins produced by the saliva. Is very nice because Rhipicephalus sanguineus and Amblyomma cageninense ticks, very common in Brazil. They produce high amounts of these molecules. These molecules are able to modulate CC chemokines and CXC chemokines. Many, the number of chemokines bound, more than 30 molecules. Most important antiques from Brazil, Rhipicephalus sanguineus and Amblyomma cageninense. So, do you remember when I said the arms race between blood-feeding arthropods and the hosts? To me, in this coevolution, the exuberance of the interaction is very strong when we try to understand in a perspective molecule of ticks, of saliva ticks, comparing with molecules from the immunity or host immunity. Only to chemokines, ticks produce a family of molecules to modulate all of these chematoctants molecules of the host. After that, I was looking for the literature, and I discovered, I'm demonstrating here, this is not an experiment made by me, but it's very nice because this experiment here was made by a group from Czech Republic, and they demonstrate for the first time to cytokine, not chemokines. They demonstrate for cytokines. They demonstrate for IL-8, IL-2, and IL-4. In a very simple experiment. We know, everybody knows about ELISA sandwich. So when we do ELISA sandwich, you get the plate, we add primary antibody, block, we add the recombinant cytokine, the secondary antibody, and review the reaction. And this group, what they do, when they co-incubate the recombinant cytokine, IL-8, IL-2, and IL-4, with saliva from different tick saliva, from ticks, Dermacentus reticulatus, Amblyomma variegatum, Ixodes ricin. They co-incubate the cytokine with saliva from different ticks. Dermacentus reticulatus is a tick from Europe. Amblyomma variegatum is a tick from Africa, but can be found in another place, but most common in Africa. Ixodes ricin is very common in Europe, and we can find also in USA. So they used saliva from many ticks from different places around the world. And when they co-incubate the saliva from these ticks with the cytokine, the ELISA was the experiment to demonstrate the detection of IL-8, IL-2, and IL-4 decrease. For some reason, some molecule in the saliva bind to the cytokine, the recombinant cytokine, and the cytokine is not detected in the ELISA sandwich. And they demonstrate this event in saliva from females and male from different species. Do you know? So they demonstrate for the first time that saliva from different ticks, from different parts of the world, are able to bind to cytokine, and this cytokine is not detected in the ELISA sandwich. And this effect is in the concentration-dependent manner from saliva of male and females, from ticks, from different parts of the world. As I said in this slide, these cytokines are very important in the immune response, IL-8, IL-2, and IL-4. But they are the same group did the same experiment to TNF-alpha. It's a very important cytokine in the inflammatory response. And they co-incubate in the same way, they co-incubate the saliva or salivary gland extract with TNF and made the same experiment, try to review the presence of TNF in the ELISA. And they demonstrate with saliva and salivary gland extract, if I increase the concentration of the saliva, the detection of TNF is decreased, is inhibited in the ELISA. They try to understand if this effect can be related with the function of TNF. They co-incubate TNF with the saliva and add it in cells, in tumoral cells. They put TNF with tumoral cells, and the TNF is able to kill cells. But if the TNF is co-incubated with saliva, if we put the saliva with in the culture, the TNF is not able to kill the cell, and the percentage of survivor cells is increased. If we increase the concentration of the saliva or salivary gland extract, the TNF lost the capacity to kill tumoral cells, showing that for some reason, molecules present in the saliva bind to TNF, and TNF lost the capacity to kill tumoral cells. It's a very nice experiment. It was made on 20 years ago. And the same group now in 2070, they demonstrated that this effect on TNF is present only to some species of ticks. They demonstrated that saliva from Ixodes ricinus, Ixodes hexagonus, Ixodes scapularis, Pessulcatus, and Hemaphysalis consina, they are the saliva of these species are able to inhibit the detection of TNF. But saliva from Dermacento marginato, Dermacento reticulatus, and Amblyomma americano does not inhibit the detection of TNF in the ELISA. So they published a table demonstrating anti-TNF active in the saliva of various species. They demonstrate that saliva from Amblyomma and Dermacento species of ticks does not modulate the TNF detection. The same thing to Hypcephalus from different species of Hypcephalus family. And we did the same experiment here. I and Anderson Samuns, we did some experiment demonstrating, in fact, that saliva from Brazilian ticks does not bind to TNF and inhibit the detection in the ELISA. But saliva from Hemaphysalis and Ixodes ticks, yes, they demonstrate capacity to modulate TNF activity in vivo, in vitro, and inhibiting the effect, the functional effect of TNF. When I came to Uberaba, I did some experiments, tried to understand the saliva from triatomines and saliva from Brazilian ticks, Amblyomma, Sculptum, Bophalus micropolus, and Hypcephalus sanguinea. Just to talk about Bophalus micropolus, all the cytokine that I have tried, no effect was found. All of the cytokines, for some reason, I don't know why, all the cytokine that I have tried to understand if they bind or not to cytokine, nothing was found until now. But saliva from amblyomma sculptum, the host tick, when I co-incubate the saliva with IL-4, IL-17, and interferon gamma, the detection of IL-4 in the ELISA is decreased in a dose-dependent manner, in a concentration-dependent manner. When I increase the amount of saliva in the co-incubation before the ELISA, the detection of IL-4 is diminished. IL-4, IL-17, and interferon gamma. And I did the same to IL-2, IL-6, IL-10, and TNF alpha. In fact, saliva does not, from this species, Hypsepis sanguineum and amblyomma sculptum does not bind to IL-2, IL-6, IL-10, and TNF alpha. As I said, saliva from both microscopic ticks, the cattle tick, does not modulate these cytokines. And I did the same with triatomines. And I didn't find any good results with saliva from triatomines, Rhodinus prolixus, Rhodinus neglectus. I didn't find the capacity of the saliva from these blood-feeding arthropods in binding to cytokines. I did some experiment trying to fractionate the saliva, and I found some fraction of the saliva that inhibit the detection of IL-4 in the ELISA sandwich. I lost the fraction. When I fractionate the saliva, I lost the fractions that maybe is able to modulate IL-17 detection, but I found also some fractions that inhibit the interferon detection in the ELISA. Unfortunately, but it's not the end, unfortunately, when I tried to find the molecules in the fraction, I lost everything. This is not the end, but it's the beginning, it's always the beginning. But when I made the studies with the professor from Butantan, Pedro Ismael, he did the fractions of the saliva, but when I go to mass spectrometry, I lost everything. So, as I said, this is not the end, this is the beginning. Here in the lab, I'm trying to enter in some area of research that is not my, I'm not expertise on this, but I'm very excited with bioinformatics and virtual screening. And I made a workflow process. Always focus on saliva from ticks. And I divide this workflow process and search of molecules in the literature for proteomes and transcriptomes. In the saliva of amblyomma sculptum, we got 80,206 proteins from amblyomma sculptum salivary glands. And we filtered these molecules using DeepLock, CD-HIT, OrthoFind, OpenFold, and AlphaFold 3, trying to understand if these molecules are secret or not, taking out all the redundancies and make the three-dimensional structure. After that, we put in the virtual screening to investigate if IL-4, interferon gamma, and IL-17 are able to bind to some of these molecules that were structured by bioinformatics. And for IL-4, they stood with IL-4. We did the same, we tried to understand the interaction of these molecules of amblyomma sculptum. And you have a positive control. When we put in this docking, when we put the IL-4 with IL-4 receptor, the binding force of interaction is 1,169. And you did another positive control, IL-4, with a monoclonal antibody. and we found 1,017 binding force of interactions between both molecules virtually during the screening, during the docking. And we found a lot of molecules from the saliva that are able to bind to IL-4. But we choose to demonstrate here the most, the 10 that had the best capacity to bind to the IL-4. So if you see here in this table, we got 10 molecules. All of them are able to bind to IL-4, and the force, the bind force by virtual screen was very similar to IL-4 to IL-4 receptor. Just to demonstrate here the interactions, here we demonstrate the IL-4 in gold, and the molecule, this molecule with this number here, and the molecule interacting with the IL-4. So we demonstrate all the interaction between the molecules. If you see here in the figure B, we demonstrate many points of this interaction that are made by hydrophobic non-covalent bonds and many others interaction that demonstrate the capacity of this molecule in binding to IL-4. We did the same to interferon gamma. We use as a positive control interferon gamma receptor interacting with interferon gamma and made the binding force using the docking and the monoclonal antibody as well. And when we found many molecules that are able to bind to TNF. And we choose one here that is very similar force of interaction when comparing interferon gamma with interferon gamma receptor. And we demonstrated as well the three-dimensional structure of the cytokine in blue. And this protein is one of the 10 in green, interacting with the cytokine. So we see many interactions that are made by IL-4 and the salivary protein, this one here. We show many interactions between these proteins. Maybe this is the best way you have to find the molecules from the saliva, because the traditional way you are trying to do from the last 20 years, it was enough to find these molecules. So we did the same with IL-17, because when we fractionate and we try when we fractionate the saliva, we lost the fraction. So when we came to the virtual screening, we found many molecules, and we choose 10 of them, and we demonstrate here the interaction was very strong. The binding force was very strong, similar to IL-17 binding to IL-17 receptor and monoclonal antibody here. So it's 2,124 binding force. They score on binding force from this document. So this is best way we found until now to discover the molecule present in tick saliva that is able to bind to cytokines. And in the end, I tried to find which family of protein this protein belongs, and three of the 10 molecules that bind to interferon gamma, 3 of them belongs to the cystatin protein or a peptidase, and the other 8 proteins are not known, and they are hypothetic. And the proteins that bind to IL-4, we found 4 of 10 that are peptidases, metalloendopeptidase, and the other 6 proteins are not known, they are hypothetic. And IL-17, we just found 1 molecule in the database. Neprilisine is a metalloendopeptidase as well, and the other 9 proteins are not yet known. As I said at the beginning, less than 5% of the molecule from the saliva of blood-feeding arthropods are known. Most of them are hypothetics. So, as I said, it's just the beginning, it's not the end. So I have many things to do, and I'm very excited and encouraged to get collaborations and what the possibilities to find by bioinformatics or by traditional way with spectrometry to find the molecules that bind to saliva. So just to conclude, uh, as I said, saliva from blood-thin arthropods are able to modulate all cells of the immune responses, all of them, from innate and adaptive immune responses. And saliva from blood-thin arthropods are able to modulate many molecules from the immune response. The, the capacity of the saliva from ticks binding to cytokine is something new, but I'm very excited that I will find something important. And we know that saliva from these tick species are able to bind to histamine, antibodies, proteins of the complement system, and many proteins are able to modulate hemostasis by modulate coagulation, vasoconstriction, and platelet aggregation. That's why I'm very excited. There are a lot of molecules that modulate chemokine. Chemokine is a cytokine. So if you know something, if you want to share something with me, some indication, please keep in contact that we can find something together. And I just published a manuscript where I just demonstrate all the molecules that bind to other molecules of the immune response. So I published this manuscript, Binding Molecules in Tick Saliva for Targeting Host Cytokine, Chemokines, and beyond. 17 molecules are modulating some molecules from the immune responses. A lot of molecules are able to bind to molecules of the immunity. So to end the presentation, I'd like to thank UFTM, my university, and Marcos Vinícius da Silva, Cioma Soares, and Virmondes Rodrigues. They are very friend and received me when I just entered in the university 15 years ago. José Marcos Ribeiro is a friend and a great master. Anderson Sanunes is a great friend and one of my, I learned a lot with him, is a great friend and a mentor. Butantan, Pedro Ismael, and many students from the lab. And most important, I'd like to thank the people that pay for the experiment in the lab. FAPMIGs, NPK, and CAPS, and the Network for Infectious Disease, Network of Translational Nanobio Platforms, Network for Biotechnology and Experimental Models, and ENCT of Molecular Entomology, that I'm very excited to work with all these people that belongs to this network and ENCT. So to end, it was a pleasure to be here, and I'm open for.
Well, thank you very much, Carlo. We're open for questions. Do we have any here? We have a question for a student here, so you want to come forward, please?
Thanks for the talk. It was really interesting. I found it really informative. So you told us that proteins from saliva were able to inhibit TNF alpha activity, which for me stood out because TNF alpha is linked to sepsis. And I was wondering if the specific protein responsible for this growth was characterized and how long it takes to promote its effects. Are there any researches aiming to evaluate its usage for therapeutic application during sepsis?
This is a nice question. As I said, the Brazilian ticks does not produce molecule that bind to TNF. For a while, we know that we have no molecule already identified that bind to TNF. But I have one result from a collaboration with UFMG, and we demonstrate using the same platform, bioinformatics and virtual screening. You have three molecules from Ixodes ricinus from USA, is a tick from Europe and USA. You have three targets already identified by virtual screening that binds to TNF. In fact, I'm very excited to understand how ticks modulate the host immune response. But in the bottom of my heart, I want to find molecules that bind to this cytokine because I want to use this like immunobiologicals. As you said, this molecule can be used to treat sepsis, for example. And I think that when you use virtual screening, we can find everything and use this in Brazil. So I have three targets that can be expressed in molecular cloning, for example, and use these molecules in model of sepsis, for example, or immune.
Do not inhibit any cytokine that you looked for. So I was wondering, because different from primates and rodents, the immune system of bovines tend to be more have a bias toward gamma delta T cells and not alpha beta T cells. So for me, that could be more permissive to the ticks, or could be necessary another mechanism, another chemokines, different molecules that we're not known yet, because gamma delta are so less understood than alpha beta T cells. What you can say about that?
No, for sure. When we talk about the immune response of cattle, we can't forget gamma delta cells. But the first impression that I have when I think about ticks and cattle bovine is because Boophilus microbes is one of the most important ticks that have a very, very specific interaction with the host. You only see Boophilus microbe ticks in bovine. For example, Amblyomma sculptum is a tick that is very common in horses. But this species is able to infest dogs, mammals, raptors, amphibians, and many other animals. So maybe during this coevolution, Amblyomma sculptum maybe was necessary to produce different kinds of molecules. Maybe Boophilus microbes only produced molecules related to the response of cattle. That's why I believe you have to find the principal cytokine or chemokine related with the cattle immune response to understand the best target. You have to find the molecules that are related with the binding activity. Maybe something like that.
Okay, thank you. And then my another question is about how the molecules are produced by the tick, because you said that in the hemolymph, in the intestine, in the body, I forget the fat body, produce the antimicrobial products, but at the salivary gland produce these kind of molecules that have a ability against the hemostasis, against the cytokines. So this could turn the salivary gland an microenvironment more prone to the development of these parasites, and this favor the infection, the cycle of the disease.
As I said in the beginning, I didn't found any effect of hemolymph, intestine, and body fat binding to chemokine and cytokines. I take out all these results because it was very big presentation. So I just said that saliva maybe is the most important tissue that produce molecules that modulate host immune responses. maybe the contact of the hemolymph or the body fat with the hosts is almost depreciated. So I believe that this coevolution brought us to a perception that salivary glands produce molecules to modulate the host response, because the contact of these molecules are very, very important. But molecules from the body fat, for example, have no a good relation to the host immune response or proximity in this interaction. That's why I believe that both of tissues are important. Both of them, body fat, intestine, and hemolymph, are important. In other perspective, they are related mainly to parasites, viruses, and protozoa, and so on. But in the context of immune response, maybe salivary glands have been demonstrated having the most important molecules.
Carlo, just to put a light in my question, I just want to understand if the tendency of the molecule production in the salivary gland can turn this more prone to the development of the parasites to be able to infect the host.
For sure, for sure. A lot of experiments have been demonstrated that saliva is able to modulate the immune response, and this is able to facilitate the pathogen transmission. There's a lot of manuscripts demonstrate saliva activated transmission. So a very simple example I can say to you is, for example, Lyme disease. The Borrelia burgdorferi, the disease is the clinical disease is strong when the Borrelia burgdorferi is transmitted by the tick infestation. The same thing happens, for example, to leishmania and the vector. If you put the leishmania with the saliva, the disease in the mice, for example, is the clinical disease is high. That's why saliva in many blood-feed arthropods are totally related with the transmission, pathogen transmission, because facilitate since the infestation and during the evolution of the disease.
Thank you. So we have a question from Bruno.
Hi, Diego. Thank you. We actually have a question from one of our students here, Clara, that unfortunately had to leave for another meeting with the graduate program. But she was wondering, when you think about co-evolution, and what would be the correct species for the cytokine that you are testing the tick saliva against? Because the lack of anti-cytokine effect on any particular tick species can be evidence of a lack of production of protein that will bind and inhibit that cytokine, but also maybe a sign of specificity of this protein against cytokines made by one particular or a narrower group of species that these ticks would eventually infest. So first part is what is the origin of the cytokine you use, and how these may affect the data that you found.
It's a nice question. The first thing that I thought when we talk about that is that saliva, the compounds of a tick saliva depend of the tick is totally different, because ticks depend of the country. They have different hosts. They have different challenges. I have one example that for me is the something that I have a delay to accept with triatomines. I did the proteome of the saliva from triatom infestans, from Rhodinus, from Mecus, and from Pastrongilo. They are 4 triatomines. Do you know? 4 triatomines. But when we published the proteome of these both triatomines, only one protein was similar in the saliva from this blood-feeding arthropod. So my perception is each one has the different challenges, different pathogens, and different mechanisms to co-evolve with the host and the parasite they are able to transmit. I think all of this together brings each one to the different arms races. But, for example, some of information are the same. For example, the size of the gland, of the salivary gland. The arthropods that has a blood feeding has the size of the gland is much high, is much bigger than others, even bigger than the blood feed arthropods.
Okay, I have another question here from Priscilla Olsen from the Federal University. She was watching us on YouTube, and she says, very interesting data, Carlo. Is there a systemic effect of the tick saliva in humans, and how could it affect viral or bacterial infection?
This is a good question. I think some bacteria use molecules from the saliva to complete the life cycle. Some molecule from the saliva is used by some bacteria and other microorganisms to not to modulate the immune response to the host, but to facilitate the biology of this microorganism. That's why I believe this vector pathogen host interaction has a lot of mechanisms. They live together from many thousands of years.
All right, thank you. I don't know if you have other questions there, Diego.
Oh, there's a question from Anderson and Gustavo too. So
thank you, Carlo. It's great to see like how your the directions you're following this work. You know, I always have to be the devil's advocate on some of these approaches. And my question to you is more like about the bioinformatics parts, like where like those analyses you've done using the ZDoc score. So how did you get, what is the threshold for that kind of assay? And I'm asking that because like when you change the cytokine and the partners, the number is very different. So if I remember correctly, you started with IL-4, the threshold was about like 1,000, and then you moved to IL-17, was totally different. And the reason I'm asking you that is because it seems to me that most of the proteins you found as potential ligands are not really ligands, but enzymes. And I'm just like wondering whether you're finding enzymes that break or to degrade the cytokines and not real binding proteins. And finally, have you used your algorithm to, have you tested your algorithm against, for example, evasins and the chemokines they bind to see if it works well, as a way to test if the software is good enough to detect binding and not like degradation sites or like, you know, enzymatic sequences. I don't know if it was clear.
Okay, I understood very well. First, we try to do everything in virtual screen using as positive control the receptor and the cytokine. And we have the first thing we did for some in some experiment was some evazines and the ligand to see if our methodology was right. And we found very good information about our experiment, because when you use the evazines, the result was similar what we found in the literature. Do you know? We found the same thing in the literature, but in that traditional way, crystallography and so on. But when we come to the experiments with bioinformatics, it's like you have to try. Do you know? You have to try. Sometimes I'm not so happy because some of the molecules, the contact is not so it's not as strong in binding force, but the structure of the molecules are not so connected. Do you know I'm trying to say? So I'm trying to increase the mechanism why I decide which kind of molecule is used to really choose the one that make a better configuration of the interaction, not only touch, but interacting in different ways to facilitate the decrease in the dissociation, constant of dissociation. Even less is better. So I'm trying to improve and find the better one that has a strong interaction in complexity. I'm not saying I'm trying to explain well, but I'm not want only interact, but I want a very complex interaction between the molecule and the cytokine. But I believe that I'm the right way. But we can do better. But I think I'm the right way, and because we demonstrate like the literature with the cytokine and the receptor, cytokine and the monoclonal antibody. We demonstrate the same thing with the monoclonal antibody and the receptor. So I think for us, only you have the option to reduce the protein and try it in vitro and in vivo. It's just the beginning.
Hi, Carlo. It's great presentation. I have a few questions, but because of time, I gonna ask just a couple of them, more like curiosity. So one thing is that you show difference between the saliva of males and females, and maybe I'm
For sure.
was not aware of the differences in terms of infestation. But some of males display better activity, and then in another part of the graph was the females. So why is that?
The general information about that is the major publication about this about the effect of saliva from ticks is females need a lot of quantity of blood. So with the manuscript already published demonstrate that saliva from females are stronger than saliva from male, because males sometimes does not need blood, but in some case yes, but the effect of the saliva is not so strong like the saliva from the females. In general terms, sometimes one point here or other is higher, but in general terms, saliva from the female has a stronger effect on cells and molecule of the immune response.
Right, but does this relate to the fact that females, infest more than males or stay longer in the
That's right,
animal?
right.
Okay.
The
And in
effect
the other
is
case, in the other case, when males has a stronger, is that correlated with the same thing, or it was just like coincidence?
my opinion is that saliva female needs to be more protected against the host immune response because females needs more molecules, because if you see a tick, a female tick, they increase in size 200 times. They increase in size because of the blood nearly 200 times. So the female needs to protect against the many cells and molecules that was ingested during the hematophagy. So they need a lot of molecules in size and quantity and complexity to modulate the host immune response, different of the male that just need a little bit when necessary.
Okay, so then it comes to my final question, is that whether the host modulates the content of saliva, because I understand that you guys just get the ticks from the whatever the lab and collect saliva in a state that they were not feeding of anything. You induces saliva, but so the thing is that if the ticks are, constantly feeding a certain host, do this modulate the expression of molecules in the saliva? And a tick that you just mentioned, Vinicius, a tick that infect only cattle, compared to a tick that infects multiple hosts, does these multiple hosts modulates the saliva of the tick?
This is nice because when I demonstrate the saliva modulating cytokines, chemokines, and many cells, most of the results are from saliva that was collected in the last 3-4 days of infestation. The ticks produce different levels in quantity and quality of bioactive molecules. The production changed during the life cycle, but most of these molecules are produced only in the last 4 days, but change during the infestation. So depends on the species of the tick, the number of molecules and the difference in the concentration and complexity of this molecule, you changing during day by day into the host.
Super. Congratulations again. It was great work and great talk. Thank you very much.
Thank you very much.
Do we have any more questions in Rio, or are we done?
No more questions here. Thank you very much.
So, all right. So thank you again, Carlo. Thank you very much. Nice seeing you again, my friend.
Bye-bye. Thanks a lot.
Bye-bye. Thank you.